Hello students, welcome to this session. Let's study structure of immune system and immune response. Immune system cells present in spleen, liver, lymph nodes, thymus, bone marrow and circulate through blood and lymphatic fluid is called as immune system. The lymphatic system it consists of thymus, lymph vessels, lymphatic tissue and lymphocytes. Thymus location it is bilobed organ present in the root of the neck above and in front of the heart. Structure: the thymus is enclosed in a capsule and divided internally by cross walls into many lobules, each full of lymphocytes (WBCs) associated with the antibody production. In relation to body size, thymus is largest organ at birth. It doubles in size by puberty, after which it gradually shrinks, its functional tissue being replaced by fatty tissue. Lymphocytes produced in thymus are T lymphocytes. Function: infancy, thymus controls development of lymphoid tissue and immune response to microbes and foreign proteins. In adult, the function is unclear. Phagocytic cells, a cell that able to engulf and digest bacteria, protozoa, cells and cell debris and other small particles is called as phagocytic cell. The types are blood macrophages. They are mononuclear lymphocytes T and B and monocytes polymorphonuclear neutrophil, eosinophil, basophil. Tissue macrophages, histiocytes. So lung alveolar macrophages, liver Kupffer cells. Function of phagocytic cell is phagocytosis. Phagocytosis is the engulfment and digestion of bacteria and other foreign particles by phagocytic cell. Mechanism: the cells move toward the foreign particle. Their abundant cytoplasm produces pseudopodia. Pseudopodia engulf foreign particles. Phagocytosed particle is held inside the vacuole is called phagolysosome. It fuses with the lysosome, forming phagolysosome. Lysosomal enzymes digest the particle, the remnants being removed from the cell. Importance: phagocytosis is effective defense against most microorganisms like bacilli of typhoid, tuberculosis, brucellosis. Neutrophils are actively phagocytic and form predominant cell type in acute inflammation. Eosinophilic leukocytes are found in large numbers of allergic inflammation, parasitic infection. Basophils contain basophilic granules, histamine, serotonin, and other hydrolytic enzymes help in digestion of the foreign particles. Let's study immune response. Immune response, the specific reactivity induced by in host by an antigenic stimulus known as immune response. The protection against invading microorganism. Reaction against any antigen. Consequences: beneficial, indifferent, injuries to the host. Highly coordinated reaction of the cells of immune system and their products. There are two types: humoral or antibody mediated immune response and cell mediated immune response. Humoral or antibody mediated immune response. So it occurs through 3-6 steps. First, activation of B cells following contact with the microbial antigen. B cells act as APC, antigen processing cells. Proliferation and differentiation of B cells into effector cells and memory cells. Third, effector function: secreted antibodies by plasma cells, which in turn counteract with the microbes in many ways, such as neutralization, opsonization, complement activation, etc. Activation of B cells. Antigens that activate B cells fall into two categories. Most antigens are thymus dependent antigens. They activate B cells indirectly via activation of T cells. The TD antigens are processed by APC, presented to T helper cells, following which the activated T helper cells cytokines produced and that in turn activate B cells. Thymus independent TI antigens, example bacterial capsule, are not processed by APC. They can directly activate B cells without help of T cell induced cytokines. Second, proliferation and differentiation of B cells. The new B cells released from bone marrow go and house in the B cell areas of the peripheral lymph nodes. Example, cortex of the lymph node and marginal zone of spleen. The new B cells are organized to form primary lymphoid follicles. Effector function of AMI promotes opsonization, transcytosis, mediates mucosal immunity. Transcytosis of IgA to gut lumen provides mucosal immunity by neutralizing the microbes at local mucosal sites. Activates complement mediated inflammation and cytolysis. Promotes ADCC. ADCC is principally cell mediated. Antibodies direct the cells to reach the target cells. ADCC is important to provide immunity against helminths, tumor cells, and virus infected cells, that is NK cells, IgG mediated. Humoral antibody mediated immunity. Primary defense against most extracellular bacterial pathogens, helps in defense against viruses that infect through the respiratory or GI tract, prevents recurrence of virus infection, participate in pathogenesis of immediate hypersensitivity and certain autoimmune diseases. Now, antibody production consists of three steps: efferent leaf, entry of antigen, its distribution and fate in tissue, contact with appropriate immunocompetent cells. Central function: processing of antigen, control of antibody production. Efferent leaf: secretion of antibody, distribution of antibody in tissues and body fluids, manifestation and effects. Cellular or cell-mediated immunity protects against fungi, viruses, and facultative intracellular bacterial pathogens. Participate in rejection of homografts and graft versus host reaction. Provide immunological surveillance and immunity against cancer. Mediates pathogenesis of delayed hypersensitivity and certain autoimmune diseases. Cells taking part in immune response, antigen presenting cells, B cells, and T cells. Production of antibodies stages: first, antigen capture and processing by APC; second, antigen presentation; third, T helper cell activation; four, B cell stimulation and differentiation; five, antibody production by plasma cells. Factors influencing antibody production: genetic factors, age, nutritional status, route of administration of antigen and site of injection, size and number of doses, multiple antigens, adjuvants, immunosuppressive agents, effect of antibody. Phases of antibody production: lag phase, no antibody is detectable in circulation immediately after antigenic stimulus. Log phase, titer of antibodies rises steadily. Plateau or steady state, equilibrium of antibody synthesis and catabolism. Phase of decline, catabolism exceeds production and titer falls. Primary immune response: the immune response after the first contact with the antigen, usually 4 to 10 days lag period. Secondary immune response: second contact with the antigen, lag period usually 1 to 3 days. Primary immune response involving B cells, naive B cells. Secondary immune response, memory B cells. Primary peak response time 7 to 10 days, secondary 3 to 5 days. Peak antibody response: primary depends on antigen, secondary 100 to 1000 times more than primary. Isotype generated: primary response, in early response IgM predominates, secondary IgG predominates. Antigens thymus dependent and independent, secondary only thymus dependent. Antibody affinity: primary lower, secondary it is higher. Immunization needed: primary a comparatively high dose adjuvant there, and secondary adjuvant not required. Primary immune response: initial contact with antigen, slow, sluggish, and short-lived. Long lag phase, low titers of antibody, does not persist for long, IgM predominant. Secondary response: response to subsequent exposure to same antigen, prompt, powerful, and prolonged. Short and negligible lag phase, very high level of antibody, lasts for long period, and IgG predominant. Here you can see the difference between primary immune response and secondary response. Cell-mediated immune response refers to destruction of cells carrying intracellular microbes and other abnormal cells such as tumor cells by various specific and non-specific cells of immune system, of which the most important is the cytotoxic T cells. Role of CMI: provides immunity against microbes residing in the intracellular environment. For obligate intracellular organisms, CMI remains the only effective immune response. For facultative intracellular organisms, bacteria like Listeria, Salmonella, Yersinia, and fungi such as Histoplasma, Cryptococcus. immunity against tumor cells and other damaged and altered cells, mediates in delayed hypersensitivity type 4 hypersensitivity reaction, plays key role in transplantation immunity and graft versus host reaction. Cytotoxic T lymphocytes, principal effector cells of CMI, involved in destruction of target cells such as virus-infected host cells and tumor cells. Naive T cells, TC cells, that is CTLs precursors, respond to viral or tumor peptide antigens which are processed by target host cells by cytosolic pathway and presented along with MSC class 1 molecules. Activated TC in turn secretes cytotoxic enzymes that lyse the target cells. Function of CTL, target cell lysis. Activated TC cells produce two types of lethal enzymes called porphyrins and granzymes. The porphyrins produce pores in the target cell membrane through which granzymes are released inside. The granzymes are serine proteases. They induce cell death by apoptosis through CAPS pathway. Second, natural killer cells, large granular lymphocytes that constitute 10 to 15% of the peripheral blood lymphocytes. NK cells are cytotoxic but antigen non-specific, part of innate immunity, act as first line of defense and do not require prior contact with the antigen. Act against virus-infected cells and tumor cells till the TC cells are activated and take over the function. Antibody-dependent cell-mediated cytotoxicity. ADCC is exhibited by NK cells secrete porphyrins and granzymes. Macrophages produce lytic enzymes and tumor necrosis factor. Monocytes produce lytic enzymes and TNF. Neutrophils release lytic enzymes. Eosinophils releases lytic enzymes and porphyrins and immunity against ailments. Induction of cell-mediated immunity, infection with intracellular microbial agents, only T cell-dependent antigens elicit CMI. The outcomes of activated T cells. First, helper T cells activate macrophages, enabling them to kill intracellular parasites. Cytotoxic T cells secrete cytokines and destroy target cells. The cytokines, first interleukins, so interleukin 1, 2, 3, 4, 5, 6, 7, 17. And then colony stimulating factors, GM-CSF, G-CSF, M-CSF. Tumor necrosis factors, tumor necrosis factor alpha beta. Interferons alpha beta gamma. Detection of CMI, mixed lymphocyte reaction, in vitro system for assessing T cell proliferation in cell-mediated response. Cell-mediated lympholysis, in vitro assay for testing cytotoxic function of effector cells of CMI. Graft versus host reaction is in experimental animals, provides in vivo system for studying cell-mediated cytotoxicity. Difference between humoral and cell-mediated immunity. Humoral mediation by antibodies, cell-mediated by cells. Cells involved humoral B cells along with T helper cells directly bind to soluble antigen. CMI T cells, CD4 Th and CD8 PC cells, T cell-mediated antigens can activate CMI. Mediators, humoral immunoglobulins IgG, M, E, A, and D. Cell-mediated cytokines, lymphokines, monokines, interleukins, and interferons. Role in complement, humoral plays role in, IgG plays role in classical pathway, CMI no role. Hypersensitivity, humoral type 1, 2, 3, CMI type 4. Immunological surveillance, humoral limited role in surveillance and tumor immunity, major role, CMI major role in immunological surveillance and tumor immunity. Transplant rejection, humoral minimal role, cell-mediated major role in homograft rejection and graft versus host reaction.
Hope you find this session useful for your understanding and for your exams. Thank you.